Welcome students. Today I want to explain some basics of imaging and related techniques. Before I go through in detail, firstly I want to show you the portion syllabus. Firstly, we will discuss the principles of microscopy, then the light microscope, microscope, confocal microscope, the flow cytometry related technique or FACS, that is the fluorescence activated cell sorter, basics of electron microscopy, transmission, and scanning electron microscopy. These things is all this thing I want to cover today. Already I provided you, all of you, the detailed course material and reference material for this portion. So first, there are various types of microscope, and if you just want to consider what a microscope performs, it's actually a device which is used to magnify small objects. So it has a capacity to magnify. And if you all want to consider its basic qualities, there are some basic features of a microscope or the technique microscopy. So what are they? A microscope. It has the property of magnification. It has a quality of resolving. Resolving power. So firstly, what is magnification? It is a quality to increase of the image in size. Like this is the original portion of a small algae, usually found as a phytoplankton, Cosmarium. Or maybe some other one. Like a dinoflagellate, which is a commonly common. Ceratium. But we can't observe them using our naked eyes. We have to study them by a microscope. So it can increase the size of the object. And we can simply calculate roughly the strength of any magnification strength of any microscope by simply multiplying the strength of ocular lens and objective lens. Suppose a particular compound microscope with a ocular lens power is 10x, and the objective lens also have the power of 10x. So it has a 100x magnification, or it can magnify arbitrarily 100 times. So this is very crucial, and this is very important to remember. Otherwise, we can also calculate the magnification by the technique micrometry. Okay. Now, another important associated aspect, that is the resolving power. This is very much associated with magnification. Resolving power actually indicates the quality of a microscope to differentiate between 2 points. How much they can differentiate? Now we can very easily differentiating. Now still also we can differentiate, but now it is very difficult to differentiate. A microscope with a good resolving power can resolve this, this, and this very clearly. The most critical condition explaining here, which is very difficult to differentiate, but a microscope with a good resolving power can differentiate this as 2 points, although they are very closely placed. So a good microscope must have a higher magnification quality with a suitable or appropriate resolving power. Now the question is how we can regulate the resolving power of a microscope. There is another important criteria known as numerical aperture. Numerical aperture. A microscope with higher numerical aperture can develop a good resolving power or higher resolving power than with a microscope with low numerical aperture. So, numerical aperture is very important to establish a good resolving power. Then another important aspect of microscopy is known as refractive index. Refractive index. Or can also call them as index of refraction. So, how we can interpret this? Actually, this is a mechanism by which we can identify the bending of a ray of light when it passes from one medium to another. This bending is known as the refractive index. So, how we can represent this? Firstly, draw this. Just consider this is vacuum and this is medium. Now this is the light. It comes from one medium and then pass into another medium. Okay. Initially, it forms an angle, and here also it forms an angle. So this is the incident light. This is the angle of incidence. This is the refracted light. And this is the angle of refraction. Okay. So this image clearly demonstrating the nature of refractive index and what actually going on in a microscope, that a light is passing from one medium to another, and depending on this criteria there are so many types of or the amount of refractive index varies from one microscope to another. So a microscope with a good refractive index produces a very good image. And to increase the quality of the refractive index, sometimes we change the mounting media, or we use some oil like immersion oil which increase the refractive index. Usually normal, if we use water, it has a refractive index of like 1.3. If we use glycerol, it has a refractive index of nearly 1.45 to 1.47. Or if we use some immersion oil, it has a very high refractive index, 1.51 and greater. So by using immersion oil, we can increase the refractive index and simultaneously the quality of the image also improved. Now, please follow the types of microscopes. What do we have to cover? So firstly, the light microscope. This is the basic one. Then we go through the phase contrast microscope, confocal microscope, fluorescent microscope, and electron microscope. This is the basic architecture of a compound microscope, what we usually follow in our practical classes. Firstly, follow this image. So, I think we all can pinpoint and identify the portions very easily. This is the ocular lens. This is objective lens. There is also a lens. This is known as condenser. Okay. This is known as rough adjustment screw. This is known as, similarly, fine adjustment screw. And we all know that usually there are 2 to 3 types of objective lens, like 10x, 40x, 55x, and in some cases 100x. And for this 100x, we have to use some immersion oils, which I already mentioned. And this immersion oil increases the refractive index. Now, this is the movable stage. Okay. And this is the light capturing device, mirror. Or alternatively, there may be a system of artificial light which can provide light for this microscopy. Now please follow this basic diagram or diagrammatic representation. This is the light source, the condenser lens, the specimen of the movable stage, objective lens, focal plane, and it forms the enlarged image. So, an image is usually formed in light microscopy by a suitable staining. It increases the quality of the sample. Along with it helps to differentiate various portions or various parts of the particular specimen of interest. Either we can stain by single stain using a homogeneous or uniform type of tissue, or we can use double stain to differentiate various types of tissue architecture. And the most important thing, what we have to consider, that by staining, we are killing the specimen. So, all the specimens, what we have to observe under this type of microscopy, are completely killed and preserved. Now, please follow other type of microscope, like phase contrast microscope, confocal microscope, etc. This is a typical phase contrast microscopy. And please consider that this phase contrast microscopy and the other types differentially abled video enabled phase contrast microscopy can very much suitable to observe, to study live cells. So in phase contrast microscopy, this is a specially constructed phase plate. And in this phase plate, in this microscopy, two things are formed. One is known as unobstructed light, phase unaltered by the specimen. So this portion of light is not altered by the specimen, this one. And the second one is known as thin line, refracted or diffracted light. This is known as phase altered by the specimen. So these two portions, unobstructed, the former one, and the obstructed or refracted light, they form an image which can differentiate by this quality, and they can reveal its cellular pattern easily and in a live condition. Similar type of microscopy known as differentiated interference contrast microscopy. They also can easily reveal the nature of a live specimen. Now, this is a basic diagrammatic representation of a confocal microscope. Now, these two things we have to consider. Actually, confocal microscope is a type of fluorescence microscopy, where we excited a specimen at its excitation wavelength, and it forms its image at its emission wavelength. Now, in confocal microscopy, only the image formed by its focal plane. Initially, it directs its image formation for a short portion of the sample. Simultaneously, there are so many slices of the sections, and ultimately the computer develops a three-dimensional constructed image of the specimen. So, in confocal microscopy, it reveals a 3D sample. Now, in fluorescence microscopy, what we want to discuss initially at this point, that in this microscopy we have to use some fluorescent compounds. This is very important to note. This fluorescent compounds, they are very important and very attractive to reveal the presence of a type of compound. And by their presence, easily we can differentiate, easily we can pinpoint their location, movement of one molecule to another, and the subsequent interaction with one molecule with another. All these things we can decipher by the fluorescence microscopy. Some basic important dye like rhodamine. Okay, it yields a red color. Then GFP, or green fluorescent protein. We get this from a jellyfish. And there are some also other fluorescently activated proteins like CFP, or cyan fluorescence protein. And the last one, YFP, or yellow fluorescence protein. So these are all various types of protein. What do we have to know to understand its mechanism? And in fluorescence microscopy, there are two specialized techniques. Okay. One specialized technique we have to study. The first one is known as FRET, or fluorescence resonance energy transfer. And the second one is known as the FRAP. And the next slide, that is fluorescence recovery after photobleaching. Now please consider this one, that is fluorescence recovery, fluorescence resonance energy transfer. By this special technique, we can explain whether one particular molecule, suppose this one, can interact with this one, and if they interact with each other, their resonance pattern or character can change, and revealed by this experiment. And second one, fluorescence recovery after photobleaching. By this technique, we can easily mention the lateral movement of any molecule. Okay, so firstly, this one, that is FRET, or fluorescence resonance energy transfer. Firstly, labeling of protein with a fluorescently tagged antibody for fluorescence microscopy. The primary antibody, this is the basic pattern of the fluorescence microscopy. The primary antibody recognizes the antigen of interest and binds to it in the specimen. So firstly, this is the immobilized antigen, a protein, and this is the primary antibody of interest, which have a specificity for this protein. Then we add fluorescently tagged other secondary antibody with the immobilized antigen. Okay. And the secondary antibody is covalently coupled to a fluorescent dye like rhodamine, GFP, YFP, CFP, etc., and so many. And the second antibody is covalently coupled to a fluorescent dye that emits light when illuminated at its excitation wavelength, what I already mentioned. Okay. And they emit light, and easily we can identify its intensity, the amount of DNA and RNA, its location, its movement, all these attributes. Now the technique FRET, or fluorescence resonance energy transfer. Just follow the illustration to determine whether two proteins interact inside the cell. The proteins are first tagged with two different variants of GFP. In this example, protein X is coupled to CFP, protein X with CFP, or cyan fluorescent protein, which is excited at 440 nanometer and emits blue light at 490 nanometer. So, excitation wavelength 440, emission wavelength 490, first one. While the protein Y is coupled to YFP, or yellow fluorescent protein, which is excited at 490 and emits yellow light at 527, much higher. If protein X and Y do not interact, illuminating the sample at 440 yields fluorescence at 490 from the CFP only. But if they are interacting with each other, this is happening. When the protein X and Y interact with each other, FRET, or fluorescence resonance energy transfer, occur. And the illuminating sample at 440 nanometer excites the CFP, or cyan fluorescent protein, whose emission in turn excites the yellow fluorescent protein, resulting in the emission of yellow light at 527. So what is happening? That initially we are exciting this protein X at its excitation wavelength 440, and as it approaches the protein Y, it is exciting at much higher wavelength, that is 527 nanometer. This is the most important expression of these experiment, that is FRET. Now, the second one, that is the fluorescence recovery after photo bleaching. This technique easily indicates whether there is a lateral movement of a particular molecule of interest. Then, this is also a very important aspect of fluorescence microscopy. Actually, this is a fluorescence activated cell sorter. Follow this. This is a fluorescently activated cell sorter. A fluorescently activated cell sorter where an antibody specific for a particular cell surface protein linked to a fluorescent molecule and then added to a mixture of cells. So there are so many types of cells, like 5 types of cells, A, B, C, D, E, with a specific character, criteria, etc. Where the individual cells pass through this laser beam, here is the laser, pass through this laser beam, they are monitored for their fluorescence quality. Droplets containing single cells are given a positive or negative charge, depending on whether the cell has bound the fluorescently tagged antibody or not. That is, not all cells like A, B, C, D, E, they all have the capacity to attach with the antibody of interest. Some of them may attach and some are not. And depending on its quality and its charged nature, they are separated into various cell collectors, like this positively charged one here, negatively charged one here, and deflected ones collected here. Okay, so in this way, by using this technique, that is fluorescence activated cell sorter, we can differentiate a specific cell or one or two types of cell from a mixture of cell. And after this separation, we can culture them or we can further analyze them for their specific character. Now, please consider another important microscopy technique, that is electron microscopy. So in electron microscopy, the most important aspect that there is no light, no glass lenses. Here electromagnetic lenses replace the glass lens. We have to prepare the sample for either the same or the TEM or transmission electron microscope. And especially for TEM or transmission electron microscope, we must have a very thin section. And as in this case, the electron beam passes through the specimen or the sample, the primary electron beam forms the image, and in TEM the image is a 2D in nature, and in SEM the secondary electron beam forms the image and they form a three-dimensional image. So initially, in both the cases, electromagnetic lenses create the image. We can increase the magnification by increasing the intensity of the electron flow. For scanning electron microscopy, in this case, the secondary electron beam forms the image. They are with a lesser resolving power than the transmission electron microscopy. For scanning electron microscopy, a heavy metal coating is very much required, usually by osmium tetroxide and by other chemicals, and they form a clear image. In this case, the primary electron beam, after falling on the specimen, and from the specimen, the secondary electron beam forms the image on the cathode ray tube. So for external surface features, usually we follow the scanning electron microscopy, and for internal ultrastructural details, we can follow the transmission electron microscopy, where the electron beam passes through the specimen, and it has a much higher resolving power and much precision. So today we have a basic idea about normal light microscope, that is represented by compound microscope, then fluorescence microscopy, confocal microscopy, and other types of electron microscopy, that is scanning and transmission electron microscopy. For reference purpose, you can follow these books, and there are so many other books also. Holmes and Hooper, Biochemistry; Wilshire Walker, The Techniques of Biochemistry and Molecular Biology; and especially in the cytology portions of the molecular biology books, and other material. So best of luck. Please go through the lecture, follow the tutorial, take care of your health and your family, especially in this pandemic situation. Best of luck.